Imagine standing in your backyard on like a really crisp autumn morning, just watching a tiny, delicate house wren picking at a bird feeder.
Yeah, a super common sight, right?
Exactly. But now, I want you to imagine that exact same terrestrial songbird ending up in the stomach of a 15-foot serrated-toothed tiger shark, hundreds of miles out in the middle of the ocean.
It sounds like a joke, honestly, or a mistake in the data.
Right, but it's not. Welcome to the deep dive. Today we are looking at how a frankly bizarre discovery, bridging the sky and the deep sea, forces us to just rewrite everything we know about the animal kingdom's food web.
It really does.
We've got a really fascinating stack of sources guiding our mission today. We're starting with some foundational university lectures on vertebrate zoology, and then layering in cutting-edge marine biology papers from biological conservation, PLOS1, and ecology.
It's a great mix of the deep history of life and very urgent modern science.
Yeah, so our mission here is to start wide. We're going to map out our broadest biological family tree and then zoom all the way in on one of its most highly optimized, misunderstood, and honestly threatened members, Cuvier, the tiger shark.
The through line here is just incredible. I mean, we are taking a journey from the ancient architectural blueprints of life stuff hidden in embryonic folds all the way to these hyper-specific, modern-day ecological crises.
Which is where that songbird comes in.
Exactly. The connection between a migrating songbird and an apex marine predator, it feels almost like a glitch in the simulation. But it actually reveals this complex, localized energy subsidy that we are only just barely beginning to understand.
Okay, but before we get out into the open ocean with the tiger sharks and the songbirds falling from the sky.
Which is still such a wild image.
Totally wild.
Huh.
But we need to understand the fundamental hardware we are working with first. We were talking about the phylum Chordata.
Right, the big biological umbrella.
Yeah, this umbrella covers roughly 40,000 species. And the range of what constitutes a chordate is just staggering. I mean, you have the sea squirt.
Oh, the sea squirt.
Yeah, which if you just look at it with the naked eye, it basically looks like a gelatinous tube glued to a rock on the seafloor.
It really does. It looks like sea trash, to be harsh about it.
Right. And then on the other end of the spectrum, you have us, humans. We're in the same club.
It really forces you to look past superficial morphology, you know? Like, despite the massive external differences between a human, a tiger shark, and a literal sea squirt, we all share three defining foundational characteristics at some point in our developmental cycle.
The entry requirements for the chordate club.
Exactly. These are completely non-negotiable. If you don't have them, you're not a chordate.
Okay, let's unpack this. The first one has to be the namesake of the entire group, right? The notochord.
Yeah, the notochord. It's basically this elastic, unsegmented cord.
I always look at the notochord like the internal steel scaffolding of a skyscraper.
Oh, that's a good way to picture it.
Because it dictates the orientation, the shape, and the fundamental tensile strength of the entire structure.
Yeah.
Like that is the underlying reason a massive shark and a human being share the exact same structural blueprint.
Biologically speaking, that scaffolding has an endodermal origin.
Meaning what exactly, for those of us who aren't embryologists?
Right, sorry. It means it develops from the inner layer of cells in the embryo. It literally pinches off from the dorsal wall of the embryonic gut.
Wow, just pinches right off.
Yeah, and if you look at lower chordates, that notochord remains their primary skeletal support for their entire life. They just never upgrade the hardware.
They just stick with the scaffolding.
Exactly. But for higher vertebrates, so including humans and sharks, it functions mostly as a temporary embryonic structure. As the embryo develops, that initial unsegmented rod is gradually replaced by the complex segmented vertebral column, the spine.
Which forms the physical armor for the second major characteristic, right? The neural tube.
Yes, the all-important neural tube.
Now, unlike the notochord, the neural tube is of ectodermal origin. So, meaning it folds inward from the outer cell layer and it positions itself right above that notochord scaffolding.
And we really can't overstate the evolutionary leap represented by the neural tube. I mean, in invertebrates, the standard architecture is a solid ventral nerve cord.
Ventral meaning along the belly.
Right, a dense, solid line of nerve tissue running right along the belly. But chordates just flipped the script entirely. We developed a hollow neural tube running along the back.
And the fact that it's hollow is the real game changer here, isn't it?
Oh, 100%. If you are relying on a solid nerve cord, you are physically capped in terms of neurological complexity. There's only so much you can do with a solid cable.
Because there's no room to grow.
Exactly. You need that hollow interior space, the neurocul, because that hollow tube is what eventually balloons outward and folds over itself at the anterior end to form the highly compartmentalized brain ventricles.
So without that specific hollow structural design, a complex brain is just off the table.
Completely off the table. The spatial dynamics of a solid cord just do not allow for the same kind of exponential expansion and infolding. It's a brilliant piece of biological engineering. It's literally what allowed for the eventual evolution of advanced cognition.
Okay, so that's 2, notochord, hollow neural tube. Then you have the third characteristic, which, well, it feels a bit more vestigial for those of us walking around on land. The visceral or pharyngeal slits.
Basically gill slits, openings in the front part of the digestive tube.
Right. And in lower aquatic chordates, those slits just hang around their whole life to house the gills for respiration.
Yeah, they use them to breathe. But in terrestrial chordates, like us, they are an embryonic signature. They show up during early development, but eventually they close up and get completely repurposed.
Repurpose into what?
Well, the tissue that forms those embryonic gill arches in humans eventually shifts and fuses to form parts of our jaw, our inner ear bones, and our throat structures.
That is wild. Our inner ear bones used to be gill arches.
Evolution is the ultimate recycler, you know.
It really is. Which actually brings us to the historical detective work that proved all this. The university lecture notes we're pulling from spent a good amount of time on this 19th century scientist named A.O. Kovalevsky.
Yes, Kovalevsky's work is legendary in this field.
Because, I mean, if you look at a sea squirt or a tunicate just sitting on the ocean floor, your first instinct is definitely not to think, ah, yes, my evolutionary cousin.
No, absolutely not. They have no notochord as adults, no hollow nerve cord, no real brain to speak of. They just filter water.
Right, so before Kovalevsky, what did scientists even think they were?
The scientific consensus was completely in the dark. They were often thrown in alongside mollusks or just grouped into these completely bizarre invertebrate categories. Nobody knew what to do with them.
But Kovalevsky changed that.
He did. His brilliance was shifting the focus away from the adult morphology. In the late 1800s, working with, honestly, relatively primitive microscopes and early embryological staining techniques, he painstakingly observed the reproductive cycle of these tunicates.
He looked at the larvae instead of the adults. He watched them before they anchored themselves to a rock and, essentially digested their own brains.
Which is exactly what they do, and what he discovered was that tunicate larvae look remarkably like microscopic tadpoles.
Oh, wow.
Yeah, and in that brief, free-swimming larval stage, they possess a perfect, unmistakable notochord. They have a dorsal hollow nerve cord, they have pharyngeal slits.
They check all 3 boxes.
All 3. They are undeniably part of our evolutionary lineage. But then, when they find a suitable rock, they anchor headfirst, resorb their tail, notochord, and nerve cord, and transition into that sessile, filter-feeding adult stage.
They just give up the higher level hardware.
They really do, but Kovalevsky proved that the developmental blueprint holds the truth of our ancestry, even if the adult organism heavily modifies it or just discards it entirely.
And the dominance of this specific phylum, this Chordata umbrella, it's just absolute. I mean, the underlying biological similarities have massive implications for our daily lives. Virtually all our domesticated animals, everything we use for agriculture, textiles, companionship, they're chordates. But the sources point out that this shared machinery also creates a pretty profound vulnerability for us.
It's a double-edged sword. We share so much cellular and metabolic architecture with other chordates that they frequently serve as highly efficient vectors and reservoirs for human pathogens.
Like diseases jumping from animals to us.
Exactly. When you look at diseases like plague, tularemia, encephalitis, or brucellosis, they are maintained in wild chordate populations, rodents, bats, ground squirrels.
Okay.
Because our biological receptors and immune vulnerabilities are fundamentally similar at a cellular level, a pathogen that evolves to hijack a rodent's cellular machinery. It often requires very little mutation to successfully adapt to a human host.
It already knows the lock. It just needs a slightly different key.
Oh.
Okay, so we have this highly scalable, incredibly adaptable biological blueprint. You can use it to build a tiny terrestrial field mouse, or you can use it to build a human.
Yep.
But a blueprint doesn't explain the timeline or the environmental pressures, right? If nature uses that exact same Corday blueprint to build a highly advanced ocean-crossing apex predator in an environment where virtually everything is trying to eat you,
Huh.
you cannot take your time growing up.
No, you really can't.
You have to grow impossibly fast.
Yeah.
But reading through the sources, it seems like for decades, marine biologists were completely in the dark about how fast that actually happens in the wild.
They were, which is the mystery tackled by the PLOS-1 research team in Hawaii. They embarked on this massive 20-year mark recapture study to establish the true growth curve of tiger sharks.
Now, I have to push back here, or at least play devil's advocate. When you first hear 20 years, the immediate reaction is like, why on earth does it take 2 decades just to figure out the growth rate of a fish? Why couldn't scientists just put them in a tank and measure them?
I get that question a lot. But captivity completely breaks the data.
Really? How so?
Captivity inherently alters every single variable. It changes their caloric intake, their metabolic expenditure, their stress hormones, their movement patterns. A captive shark is just not a wild shark.
That makes sense. It's like trying to study a wolf's hunting stamina by watching a dog in a kennel.
Great analogy. To get ecologically relevant data, you absolutely have to measure them in the open ocean. But the logistics of tracking a highly mobile pelagic predator that can literally cross ocean basins, they are staggering.
It sounds like a brutal physical grind. I mean, the researchers in Hawaii were setting demersal longlines, bringing these massive animals alongside a small 6-meter skiff in rolling Pacific swells. We're talking about tail roping a thrashing, potentially 15-foot shark and physically inverting it in the water to induce tonic immobility.
And tonic immobility is such a fascinating physiological response.
Yeah, explain how that works, because it sounds like magic. You just flip a shark upside down and it goes to sleep.
Essentially, yes. By turning the shark upside down, you overstimulate their sensory apparatus, particularly the ampullae of Lorenzini.
Those are the little pores on their snout, right?
The electroreceptor network. This massive sensory overload triggers a reflexive paralysis. It essentially puts the shark in a temporary catatonic trance. It lowers their respiration and allows the researchers a brief window to safely work without the animal thrashing around and injuring itself or, the crew.
Okay, so in that tiny window where the shark is hypnotized, they take their measurements, pre-caudal length, fork length, total length. They insert a tag into the dorsal musculature, flip the shark back over, and let it swim away. And then they just have to wait and hope they somehow catch that exact same shark again, maybe months or years later, in the vastness of the Pacific Ocean.
It's like finding a needle in a haystack, if the needle was swimming across the Pacific. Over 20 years, they tagged 420 sharks, and they managed to recapture exactly 50.
50 out of 420 over 2 decades. It's like trying to measure the height of a runaway teenager who only comes home for dinner once a year and half the time your ruler is broken.
That is exactly what it feels like. And historically, the scientific community tried to bypass this logistical nightmare by relying on a completely different method, counting the growth rings on a shark's vertebrae.
Oh, like counting the rings on a tree.
Exactly the same basic principle, dendrochronology, but for sharks.
But the PLOS-1 paper calls out the inherent flaw in that historical methodology, right? They specifically label them unvalidated vertebral ring counts. The assumption was that a shark laid down one distinct calcified growth ring on its vertebrae every single year.
Yeah, and the problem lies in the underlying biological mechanism of calcium deposition. Does a ring always equate perfectly to one solar year?
Probably not, I'm guessing.
Right. What happens if the animal experiences a prolonged period of starvation or a massive spike in stress? Those environmental factors can cause irregularities in calcification, creating false rings.
So you think a shark is 10 years old, but it's really only 5. It just had a really stressful couple of years.
Precisely. Furthermore, as sharks reach maturity and their growth naturally slows down, the wings become incredibly compressed and almost impossible to distinguish. Without empirical validation, catching a wild shark, measuring it, letting it grow for a known period, and measuring it again, those vertebral age estimates were just deeply flawed.
So what did the new empirical mark recapture data actually show?
It completely shattered the old assumptions. The math revealed that tiger sharks in Hawaii are growing twice as fast as the unvalidated vertebral count suggested.
Twice as fast.
Yes. The researchers found that, on average, a tiger shark reaches roughly 11 feet in length, about 340 centimeters, by the time it is just 5 years old.
That is an unbelievable amount of mass to put on in 5 years.
It really is. And to process that kind of extreme biological variance and the inevitable measurement errors, the researchers had to utilize a highly specific statistical framework. It's known as the Francis Maximum Likelihood Model, or GROTAG.
Okay, wait, GROTAG. What does that actually do for the data? Because if you're measuring a 12-foot shark on the side of a rolling skiff, your tape measure is definitely not going to be perfect to the millimeter.
Exactly. And beyond the measurement error, you have the reality that animals are not manufactured on an assembly line. One shark might have found a highly productive hunting ground and gorged itself, while another struggled to find food and grew slower.
Right.
So the GROTAG model is essential because a simple average would distort the reality of population. Maximum likelihood estimation is a statistical philosophy that weighs the probability of the observed data against various expected growth curves.
So it filters out the noise.
It systematically isolates the noise of measurement error, and it accounts for individual biological variability differences in genetics or localized resource abundance. By crunching all those variables, the GROTAG model outputs a highly refined representation of the population's true growth trajectory. Biologists refer to it as the von Bertalanffy growth equation.
And does that model also project like the absolute maximum sizes we can expect these sharks to reach?
It does. It estimates a maximum average size of just over 13 feet, around 403 centimeters.
But nature always produces outliers, right?
Always. The absolute largest shark recaptured in the Hawaii study was a massive female measuring over 15 feet long, 464 centimeters.
15 feet. That is a submarine. And it brings up a really crucial biological characteristic mentioned in the data.
Yes.
Sexual dimorphism.
Yes.
In the catch data, researchers documented significantly more females than males, and the females were consistently much, much larger. Why is that?
For tiger sharks, massive physical bulk in females is an evolutionary necessity. They are viviparous, meaning they give birth to live young. And a female tiger shark can carry a staggering litter of 30 to 50 pups at a time.
50 pups.
Yeah. She requires a massive internal body cavity simply to accommodate that kind of reproductive volume, so they have to get huge.
The tagging data also highlighted just the sheer scale of their environment. I mean, they recaptured one specific tagged shark, not in the waters around Hawaii, but in the Gulf of California, off the coast of Mexico.
It's mind-blowing.
That is a straight-line migration of roughly 5,000 kilometers in a single year. These aren't just local predators. These are transoceanic predators utilizing the entire Pacific Basin as their hunting ground.
They are. But returning to that staggering growth rate for a second, packing on over 3 feet of mass a year as a juvenile, it demands an explanation. Why would an organism pour such an overwhelming amount of metabolic energy into rapid physical expansion?
Well, it's a desperate race to age out of the prey category, isn't it?
Exactly.
When a tiger shark is born, it drops into the ocean at under 3 feet long. And in the pelagic zone, the open ocean, a 3-foot animal is universally viewed as just a protein source. Right, everything wants to eat it, including larger adult tiger sharks. The ocean does not offer safe nursery pens. So growing incredibly fast is not a luxury, it is their primary survival strategy. They have to get big, and they have to get big immediately.
But rapid growth requires fuel. You do not reach 11 feet in 5 years without a massive, consistent intake of high-density caloric energy. The metabolic demands of that growth curve are astronomical.
Which brings us to one of the most unexpected discoveries in recent marine biology, detailed in our sources from the journal Ecology. Because this desperate caloric need drives these fast-growing juveniles to exploit a deeply, deeply unusual food source.
Yeah, so for this, the researchers shifted focus from the Pacific to the Gulf of Mexico, specifically the coastal waters off Mississippi and Alabama. They were conducting dietary analyses on 105 tiger sharks over an 8-year period from 2010 to 2018.
And the methodology here involves something called gastric lavage for the live sharks, right?
Yes, gastric lavage. It's essentially pumping a controlled volume of water into the shark's stomach to flush out the contents so they can analyze them, and they also examine the digestive tracts of deceased specimens.
And the findings were completely counterintuitive. Out of the 105 sharks analyzed, 39% of them, so 41 sharks, had bird remains in their stomachs.
Now, if you ask anyone to picture a marine predator eating a bird, the mental image is always an albatross resting on the ocean swells, or maybe a pelican diving for bait fish. You expect an interaction between two marine species.
But the researchers found 0 marine birds, not a single gull, pelican, or albatross.
Zero.
Through visual identification of the recovered feathers, which, surprisingly, actually survived the initial stages of stomach acid, and by utilizing DNA barcoding, they identified 11 distinct species of terrestrial birds.
Land birds.
Land birds. We were talking about the eastern kingbird, the barn swallow, the house wren, the yellow-bellied sapsucker.
And just a quick note on the DNA barcoding, it's such a fascinating tool in dietary ecology. When visual identification is compromised because the animal is, half digested, researchers isolate a specific genetic sequence. Usually it's the cytochrome secus oxidase subunit I gene, or COI gene.
Right.
They amplify it and run it against a global genetic database. It leaves absolutely zero room for ambiguity. These sharks were definitively consuming small terrestrial songbirds.
But the visual of a 15-foot apex marine predator snacking on a tiny yellow-bellied sapsucker is absurd. As you said earlier, it feels like a glitch in the ecosystem. How does a terrestrial songbird end up in the digestive tract of a pelagic shark?
So the researchers actually solved this mechanism by correlating their stomach content data with massive data sets from eBird.
eBird, that's the citizen science project tracking bird movements.
Exactly. They looked at the exact dates they were finding these terrestrial birds and the sharks, and they aligned those dates with the migratory patterns being logged by bird watchers on the coast.
And the alignment was flawless. Because every fall, billions of birds embark on this massive migration from North America down to their wintering grounds in Central and South America. And for many of these species, the most direct route requires a continuous flight straight across the Gulf of Mexico.
It is a phenomenal test of endurance. A tiny songbird is pushing its metabolism to the absolute limit to make that trans-Gulf crossing. To survive, they rely on favorable meteorological conditions. They essentially wait on the northern coast for cold fronts moving south, utilizing the strong tailwinds to subsidize the energy cost of the flight.
But the Gulf of Mexico is notorious for unpredictable, violent weather. If a severe squall or a sudden shift in wind direction hits while these birds are halfway across the water, it becomes a meteorological trap.
They are already operating at the ragged edge of their physical capacity.
Right, so when the weather turns against them, they simply run out of energy. Thousands of exhausted songbirds are forced out of the sky and just crash into the ocean.
And this exposes the fatal difference in anatomy between a marine bird and a terrestrial songbird. A marine bird has webbed feet and a uropygial gland that secretes waterproofing oil over its feathers. If a duck or a gull gets tired, it lands on the surface, rests, and takes off again.
But a songbird can't do that.
No. Terrestrial songbirds lack both the waterproofing and the anatomical structure to launch from the water. The moment a house wren hits the surface of the Gulf, its feathers instantly waterlog. It cannot float and it cannot fly. It simply drowns.
And this localized, weather-driven phenomenon creates what the researchers termed a windfall of nutrients from the sky. You have these massive episodic events where thousands of easily accessible calories literally rain down onto the ocean surface. It is a highly predictable, seasonal energy subsidy, and the tiger sharks are just waiting.
They are. But the most revealing piece of data from the ecology paper is exactly which tiger sharks are capitalizing on this windfall. Nearly half, 46% of the sharks with bird remains in their stomachs were neonates. Newborns.
It perfectly connects back to the growth data from Hawaii.
Yeah.
These neonates are in a desperate, biologically mandated race to pack on mass. But active hunting in the pelagic zone requires an immense caloric expenditure.
And the success rate for a newborn shark is incredibly low.
I compare these neonate sharks to human toddlers. They have very low predatory efficiency. They literally don't know how to hunt yet. They haven't developed the skills to consistently run down fast-moving fish or heavily armored sea turtles.
So they are operating as deep-sea vultures. Scavenging is a highly optimized energy strategy. Why burn massive amounts of calories attempting to hunt elusive prey when a localized weather event periodically drops a high-yield caloric buffet directly onto the surface of the water?
Like a toddler eating Cheerios off the floor. It requires zero hunting energy to consume a drowned songbird.
Exactly. These exhausted, drowned songbirds are essentially subsidizing the most vulnerable life stage of the tiger shark. The neonates are exploiting this highly specific terrestrial energy transfer to fuel the explosive growth they need to survive their fragile early months.
It is an incredibly delicate, perfectly synchronized ecological loop. Think about it. A cold front over Canada pushes a barn swallow over the Gulfs, a squall forces it down, and a newborn tiger shark uses that exact packet of calories to grow large enough to avoid predation.
It is a true testament to the profound interconnectedness of the biosphere. But on the flip side, it also highlights the extreme fragility of the system. Nature provides this narrow, precarious window for newborn sharks to survive. However, as we shift our focus to the data coming out of the Arabian Sea and the Western Indian Ocean, we see how human industrial activity is systematically collapsing that survival window.
Yeah, let's look at the research published in Biological Conservation. This paper focuses heavily on the Cochin Fisheries Harbor on the southwest coast of India. The researchers spent 16 months painstakingly analyzing the length-frequency data of tiger sharks landed at the harbor. They measured over 600 individuals.
629 to be exact.
Right. And the resulting data is a massive biological red flag. Almost 95% of the tiger sharks brought to market were immature.
95%. I mean, that figure means the fishery is almost entirely supported by individuals that have not yet contributed a single offspring to the next generation. The researchers documented the average length at first capture, which is the size at which these animals are being removed from the ecosystem by fishing gear, at roughly 171 centimeters, just under 6 feet.
Which is devastating when you look at their biological benchmarks. A tiger shark does not reach sexual maturity until it hits roughly 9 feet, or 270 centimeters in length. The overwhelming majority of the harvest is being pulled out of the ocean years before they have the physical capacity to reproduce.
And the data reveals an even darker layer. Nearly a quarter, 23% of the total landings were classified as young of the year, sharks under 5 feet in length.
The neonates.
The neonates. The exact same highly vulnerable demographic that we just established relies on seasonal weather anomalies and floating songbirds just to survive their first year. In the Arabian Sea, almost 25% of the catch consists of these helpless newborns, frequently hauled up as unintentional bycatch in massive mechanized gill nets or on long lines.
The researchers quantified the severity of this collapse using something called the exploitation rate. Without getting bogged down in all the math, the exploitation rate currently operating in the Arabian Sea completely eclipses the maximum sustainable yield threshold.
Right, the exploitation rate, or E, is at a staggering 0.71, while the maximum threshold is 0.551. They are operating far, far beyond the absolute red line where the population can theoretically, mathematically replenish itself.
So what do we call this?
When 95% of a harvest consists of immature individuals, biologists classify the situation as recruitment overfishing. You are not just skimming the surplus adults off the top of the population, you are systematically severing the pipeline of juveniles before they can recruit into the breeding stock.
Okay, so what does this all mean? Because a logical counterpoint might be to look back at the Hawaii data. We know tiger sharks possess an incredibly steep growth curve. If they pack on mass rapidly and reach maturity relatively quickly for a large animal, shouldn't they be highly resilient to fishing pressure? Fast-growing species typically bounce back.
That assumption holds true for many teleost fishes, bony fishes that release millions of eggs at a time. But tiger sharks possess a biological Achilles heel. They exhibit classic K-selected species traits.
Meaning what?
Meaning, while the individual organism grows rapidly, the reproductive cycle of the population is incredibly protracted. Female tiger sharks operate on a biennial and sometimes triennial reproductive cycle.
Meaning a mature female only reproduces every 2 to 3 years.
Correct. She invests massive biological resources into carrying a large litter of highly developed pups, but the gestation period is exceptionally long, and the subsequent recovery period before she can breed again takes years. So the mathematical reality is inverted. The individuals grow fast, but the population replenishes at a glacial pace.
So the math just doesn't work.
Not at all. When you subject a species with a triennial reproductive cycle to an exploitation rate that removes 95% of the juveniles, complete population collapse is a mathematical certainty.
But we have to look at the socioeconomic reality driving this math, too. The biological conservation paper specifically highlights the Thuthor fishers. This is a highly organized, industrialized fleet utilizing mechanized multi-day trips lasting upwards of 45 days at sea. Sharks are not just an incidental catch for them, they are a vital commodity.
Absolutely.
The fishers are operating in a globalized economy, harvesting a resource to ensure their own survival and maintain their livelihoods. So, neutrally speaking, if these fishers rely on sharks for income, isn't catching 95% immature sharks a guaranteed way to destroy their own future livelihood? If there are no adults to breed this year, there will be no juveniles to catch next year.
It's the ultimate manifestation of the tragedy of the commons, which is a structural economic trap described by Garrett Hardin in the mid-20th century. The ocean represents an open-access common resource. If an individual fisher on a multi-day trip pulls up a juvenile tiger shark, they face a choice. They can release it, allowing it to grow and potentially reproduce.
Right.
But there is absolutely zero structural guarantee that a different vessel won't simply catch that exact same shark the following day and sell it.
So the economic incentive structure just rewards immediate extraction. If you leave the resource in the water, your competitor takes the profit. The rational choice for the individual in the short term inevitably drives the collective toward long-term ruin.
And the complexity of managing this trap is compounded by the spatial dynamics of the animal itself. We know from the acoustic and satellite tagging data in the Pacific that a single tiger shark can easily migrate thousands of kilometers across international boundaries in a single year.
Which renders localized, isolated fishing bans almost entirely useless. I mean, if one nation completely outlaws the harvest of tiger sharks within its exclusive economic zone, but the sharks simply migrate across an invisible line into international waters or into the jurisdiction of a neighboring country with zero regulations, the population will continue to plummet.
The authors of the biological conservation paper recognize this inherent complexity. I mean, global tiger shark populations are currently listed as near threatened, but the localized data from the Arabian Sea indicates a staggering population decline of 30 to 50% over just the last 5 decades.
That is a massive drop.
To reverse this trajectory, the researchers propose 3 highly targeted, biologically informed management strategies designed to intersect with the animal's life cycle without immediately bankrupting the local fleets.
Let's go through them. The first major strategy involves identifying and establishing important shark and ray areas, or ISRAs. They specifically highlight the Fuvahmulah Atoll in the Maldives and a geographic feature called the Waj Bank off the southern coast of India.
And the selection of those specific geographic coordinates is driven entirely by the biology. The ocean is not a uniform expanse of water. It contains critical infrastructure. The Fuvahmulah Atoll and the Waj Bank are recognized both by empirical acoustic data and by generations of local fisher knowledge as vital reproductive sites. They are popping grounds.
Safe zones.
Exactly. By designating these highly specific zones as protected no-take areas, even if only temporarily during the pupping, season management can offer a localized sanctuary. It provides a spatial refuge where pregnant females can safely give birth and where the neonates can spend their most vulnerable early months without encountering a wall of mechanized gillnets.
Okay, the second strategy tackles mechanics of the harvest itself. The researchers propose strict gear restrictions, specifically mandating larger mesh sizes for the gillnets and imposing rigid limits on the number of hooks deployed on long lines. And crucially, they suggest enforcing these restrictions during a highly specific temporal window between February and July.
Because that temporal window aligns perfectly with the reproductive timeline in the eastern Arabian Sea. Gestation typically begins in January, culminating in parturition birth in May of the following year. However, researchers documented the capture of young-of-the-year sharks as early as February, likely representing migrating populations from the broader western Indian Ocean.
So by tweaking the gear, you let the babies go.
By mandating larger mesh sizes, specifically during the 6-month window, you physically engineer a solution to the bycatch problem. A larger mesh allows the small, newly born neonates to simply swim through the net unharmed, while still allowing the fishers to target larger, mature teleost species or adult sharks that have already entered the breeding pool.
It is a practical, mechanical intervention. I like that. The third proposed strategy is the introduction of a minimum legal size limit along the Arabian Sea coast of India. They recommend setting the legal threshold at roughly 9.5 feet or 290 centimeters.
And the biological logic behind that specific measurement is just ironclad. We know the average size and sexual maturity is roughly 9 feet. By setting the legal extraction limit just above that biological benchmark, you mathematically guarantee that every single tiger shark legally brought to market has already had the opportunity to complete at least one reproductive cycle.
So it's a direct structural safeguard against both growth overfishing and recruitment overfishing.
Exactly.
What makes this paper so compelling is its pragmatic approach to the human element. The researchers explicitly warn against the implementation of sweeping blanket bans on shark fishing.
Because history consistently demonstrates that blanket bans in artisanal and commercial fisheries frequently backfire. When you abruptly criminalize the primary source of income for a coastal community without providing viable economic alternatives, you do not stop the fishing, you simply drive it underground.
Right, it fuels black markets.
Black markets, widespread noncompliance, and an explosion of illegal, unreported, and unregulated fishing. Once the harvest moves into the shadows, scientists lose access to the data, and management becomes utterly impossible.
So the paper argues heavily for participatory research and deep community engagement. The scientists and policymakers have to collaborate directly with the Thuthor fishers. The management strategies must be co-developed, acknowledging the fishers' traditional ecological knowledge alongside their economic realities.
Synthesizing the data across all our sources today reveals a remarkable continuity. The conservation strategies proposed for the Arabian Sea, protecting the pupping grounds at the Wadj Bank and implementing mesh size restrictions, are ultimately designed to protect the neonates.
The babies.
Yes, it is a massive multinational effort to shield the exact same vulnerable, fast-growing newborns that we watch relying on cold fronts and drowned songbirds in the Gulf of Mexico. The policy recommendations are directly dictated by the deep evolutionary constraints of the animal's biology.
I look at the extraction data from the Cochin Harbor, like an industry clear-cutting a forest of saplings before a single tree has had the chance to drop seeds. You extract a small amount of value today, but you guarantee a completely barren landscape tomorrow. Protecting the pupping grounds and enforcing a minimum size limit is the equivalent of building a fence around the nursery. It protects the initial investment, allowing biological capital to mature and reproduce.
It is the only mathematical way to ensure the forest remains intact for the next generation of fishers. It requires a fundamental shift in how we manage common pool resources. We just have to align our economic extraction with the unyielding mathematical reality of the species' reproductive cycle.
Which brings us to the end of our deep dive today. We started by mapping the fundamental architectural blueprint of all chordates, the ancient scaffolding of the notochord, and the hollow neural tube that connects us humans to the simplest sea squirts. We explored how the open ocean pressures that blueprint to build a tiger shark, forcing an apex predator into a desperate race of rapid, mathematically verifiable growth. We uncovered their bizarre, highly localized reliance on terrestrial songbirds forced out of the sky by violent weather. And finally, we confronted the immense complexity of managing human industrial activity in the Arabian Sea, trying to halt the systematic extraction of these animals before the population crosses a point of no return.
The scope of the ecology is vast, and I would leave you with one final thought regarding the sheer scale of that interconnectedness. Think about the invisible, almost unimaginable physical threads connecting a tiny yellow-bellied sapsucker navigating a cold front in the autumn sky to the survival of a 15-foot apex predator patrolling the deep ocean. When human activity pulls on one string in the natural world, whether we are fundamentally altering atmospheric weather patterns that govern avian migrations, or dropping mechanized long lines into a pupping ground off the coast of India, we are tugging at the entire fabric of the globe. The next time you see a small bird resting in your backyard, ask yourself, what hidden ecological chain reaction might this tiny creature be a part of?